Okay, welcome back to part 2 of chapter 4 on the topic of the cells. So in part 1, we got done by talking about the different parts of the prokaryotic cell and some of the different ways, different types of microscopy we are able to use to visualize specific parts of the cell. And now we can move on to the eukaryotic cell and some of the detailed features and structures that are existing within eukaryotic cell. So, as we have mentioned, eukaryotic cells is the other group of cells where we define them as having membrane-bound nucleus or organelles and also several rod-shaped chromosomes. So instead of nucleoid, they have a nucleus that contains the chromosomes or DNA within it. And we typically divide eukaryotic cell into either an animal cell or a plant cell, where there are more significant differences between these two specific types of cell. All right, so let's go over the major structures and organelles within the eukaryotic cells, and then we will mention those that are only present in plant cells as well. All right, so as we have mentioned, all cells have a plasma membrane around them, and then the plasma membrane, as I've briefly introduced in chapter 3, is made up of phospholipids, and that they are laid out in 2 layers, which is why we call it a lipid bilayer. Okay, where the hydrophobic tails right here are facing toward the middle of the lipid bilayer, and the hydrophobic heads are either facing the exterior part of the cell in this region or interior part of the cell in this area. Okay, and within the plasma membrane, they're usually embedded with proteins for specific functions we'll talk about in the next chapter, chapter 5. And the plasma membrane's main function is to separate the internal contents from the surrounding environment. Okay, kind of acting like a barrier, because if anything and everything can go in and out of the cell whenever and however they want, a cell actually cannot survive that way, because things will leak out. Important for the cell will leak out, and things that we don't want coming into the cell will just come in whenever they want. Okay, and that can actually sometimes overwhelm the cell, and the cell can burst apart. Okay, so that is something we will discuss in the next chapter when we talk about diffusion and osmosis, how plasma membrane is able to regulate that. Okay, it can control what is allowed in and what exits out of the cell, like I was saying. Okay, in terms of ions, water, oxygen molecules, and organic molecules. Now, this feature will become an important concept in anatomy and physiology, where specialized cells in the body that performs a lot of absorption or secretion of fluids tend to have these finger-like projection folds on their plasma membranes called microvilli. Okay, so instead of being flat like what we usually see cells as, kind of flat like this with the nucleus on there, it's flat on this side, it's flat on that side. Okay, but for those cells with microvilli, one side of their cell will have these curving lines, sort of like this. Okay, with the nucleus in the middle, and that's what's depicted in this particular image here with the nucleus, and those are those curvy lines on top that are called microvilli, and they are for the purpose of providing more surface area for maximizing absorption and secretion. Okay, because when we're talking about absorption, we're talking about usually absorption of important things. Okay, and sometimes we want to have more space. The more surface area we have, the more absorption can happen. So that's why, for example, in our intestines, there are many cells that are aligned with these microvilli, because that's the main area of the body that enables for nutrient absorption from the food that we eat. Okay, if we cannot absorb whatever nutrients we eat, we will starve to death. Okay, because none of our cells are being fed with all these important nutrients that we're ingesting into our body. Okay, but because of the way that our small intestine cells are designed, we can maximize the absorption of those important molecules we're ingesting, like vitamins, amino acids, etc. Glucose, okay, all those macromolecules we were talking about earlier. All right, so the next thing we have in all eukaryotic cells is the cytoplasm, which, like I said, is the cell's entire region between the plasma membrane and the nucleus, okay, or the nuclear envelope to be more exact. So all these space area within the animal cell or the plant cell is called the cytoplasm. And they consist of organelles that are suspended in a gel-like substance called the cytosol. Okay, so make sure you understand the difference between cytoplasm and cytosol and cytoskeleton. Okay, so students always get confused with these things. Cytoplasm is just referring to a compartment within the cell. Okay, within inside of the cell. Cytosol is the actual gel-like matrix that's suspending everything within the cell. And then cytoskeleton are sort of these lines or fibers within the cell that kind of crisscross all over the place to support the cell structure. Okay, and we'll talk about that in the last part of the chapter when we get to the cytoskeleton. Okay, and the different type of cytoskeleton are there for different types of cells. So we'll move on to the next organelle in all eukaryotic cells, which is the nucleus. Very important organelle. It is usually around the center of the cell, and it houses the cell's genetic material in form of DNA. And why DNA is important, or genetic material is important, is because it directs the production of proteins using ribosomes. Or the very production of ribosomes itself is required, does require instructions from your DNA. Okay, without ribosomes, you don't have factories. You have nothing to produce proteins from. So that is why, genetic material is so important, because it regulates how everything is built within the cell. So, for the nucleus, there are several parts to it as well. Okay, so we have the nuclear envelope, which is the outer double membrane that surrounds the whole nucleus there. Okay, so just like how the plasma membrane has a phospholipid bilayer, the nucleus or nuclear envelope also has a phospholipid bilayer that separates the genetic material from everything that surrounds the cell. So inside the nucleus is the genetic material, and it separates it from all the busy stuff inside the cell. Now, around the nucleus, as we have saw in the TEM image I showed you earlier, it's actually studded with nuclear pores. Okay, so it's a little sphere that has little holes all over the place, okay, called nuclear pores, that allow some stuff to come out of the nucleus, because there's two very important things that need to come out of the nucleus on a daily basis. Okay, one of those is ribosomes. Okay, ribosomes are actually generated within the nucleolus, as we will learn in a bit next. And then there's also messenger RNA, which is a copy of the genetic material from inside the nucleus, and it has to travel to the cytoplasm in order for the protein production to begin. So we'll talk about that when we get to protein synthesis, but those are the two things that needs to come out of nuclear pores on a daily basis, is the ribosomes, okay, and the messenger RNA. And also there can be ions and other molecules that can pass between the nuclear pores as well. Okay, and what's within the nucleus, just like how we have the cytoplasm, inside the nucleus there is nucleoplasm as well. Okay. All right, and then you have the nucleolus, which is this small little concentrated circle right there. Okay, so inside the nucleus, okay, there's usually a darker dot within the nucleus. That little dot there is the nucleolus, which serves for the function in the assembly of ribosomal subunits, which I'll get to their function in a bit. But, kind of like what I said, ribosome is for protein synthesis. So where ribosome themselves are generated are within the nucleolus, and they will then be transferred into the cytoplasm for their, for their job within the cytoplasm of producing proteins. And lastly, the last part in the nucleus are the chromosomes, which are pretty much just a condensed version of the DNA. DNA can exist as a uncondensed version, kind of hanging around like that within the nucleus, or when they get ready for cell division that we'll talk about in another chapter on mitosis, DNA actually needs to be condensed into chromosomes. Okay, so chromosomes are just condensed form of DNA molecules. Okay, so they can be easily maneuvered around the cell during cell divisions. All right, so ribosomes are just then tiny dots of proteins that are either floating around in the cytoplasm or attached to another organelle called the rough endoplasmic reticulum, okay, which is what we know as rough ER. The ribosomes are actually made up of two subunits, one large subunit and one small subunit. Very easy. We named that as that, one large subunit and one small subunit. And they serve the very important function for making proteins. Okay, so they're important for protein synthesis. Now, they will specifically use two types of RNA, messenger RNA and transfer RNA specifically, to produce proteins, okay, and they will produce proteins in a way that it only adds a specific order of amino acids in a peptide chain, okay, kind of what we talked about in chapter 3 is that remember I said the structure drives the function of the protein, and what drives the structure or three-dimensional structure of the protein is the sequence of amino acids that's in the primary level of structure of protein. So what determines that is the mRNA, and then the tRNA will bring in the specific amino acid to string them up together by production of a peptide bond between the amino acid to give you a growing peptide chain. Okay. All right. Cells that usually need to produce a lot of proteins are abundant in these ribosomes. For example, in pancreatic cells, they need to, produce a whole bunch of different types of digestive enzymes that will be released into your small intestines to help with the digestion or breakdown of macromolecules, okay? Sort of like we ingest these polymers of carbohydrates, lipids, and everything into our digestive system. Well, our digestive system needs to be able to break that back down into monomers so we can be absorbed back into our bloodstream and serve as building blocks for other stuff in our bodies. So what enables that breakdown of polymers into monomers are enzymes, and those specific enzymes are created or produced by pancreatic cells, okay, particular parts of the pancreas. Different cells from what makes insulin and glucagon, though. There are two main types of cells in the pancreas. One group of cells makes insulin and glucagon for hormone, regulating blood sugar levels, but there's also different type of pancreatic cells in a different population that will just for the purpose of producing digestive enzymes, okay? All right, so the mitochondria is the next organelle we'll discuss, okay? And it is known as a cell's powerhouse because they are responsible for making ATP using our famous reaction called the cellular respiration for breaking down glucose and turning them into ATP that we have discussed in the last chapter, in chapter 6 of metabolism. So this is why you may have heard the term mighty mitochondria, is because they are mighty because they power our cells by making ATP. Okay, so mitochondria is usually abundant in muscle cells because our muscle cells are constantly moving They constantly need ATP in order to produce a movement. Okay, for example, just you holding your pen or pencil right now, jotting down notes or swiping through slides. Okay, those use a lot of ATP in your finger, your hand, and arm muscles in order to provide you with the ability to do those movements. Okay. And then the mitochondria is actually a special organelle, okay, kind of in a sense that it has not just one membrane, but bound by two membranes, okay? So it's double membrane. So there's one layer of membrane around the outside and then another layer of membrane on the inside, okay? So there's an outer membrane and an inner membrane. And we'll talk about the significance of these two types of membrane when we get into the process of making ATP in the cellular respiration chapter. The mitochondria also tend to contain its own ribosomes and DNA as well, which is why now there's such thing as sequencing or matching your mitochondrial DNA, because supposedly mitochondrial DNA only comes from the mother, because we inherit those organelles from the egg cell. All right. So the inner folds of the mitochondria are called crista, or plural form cristae. Okay, so these folds right here we're talking about within the mitochondria, those are crista. And the area surrounded by these folds is what we call mitochondrial matrix. Okay, make sure you remember these terms in terms of outer membrane, inner membrane, crista, mitochondrial matrix. Though these terms will come back to you when we talk about the different roles they play in cellular respiration, okay, the production of ATP. All right, so the next organelle we'll talk about is called peroxisome. Peroxisome are small little membrane-bound spheres that float around the cytoplasm in the eukaryotic cell, working for the purpose of breaking down fatty acids or amino acids. Usually they are no longer useful for the cell, and they also detoxify many poisons. Okay, so easy way to distinguish the function of peroxisomes from the other organelles is it starts with a P, so you can remember that as for poison, and peroxisome just sounds kind of like detox. Okay, so detoxify poisons or break down fatty acids and amino acids. Okay, and usually the liver cells would have a lot of peroxisomes where they can detoxify alcohol that the human body ingests. Okay. All right, vesicles and vacuoles are special organelles that function for mainly storage of some sort or transport of some sort. Now, we'll begin by talking about vacuoles first. Vacuoles are only present in plant cells, okay, not in animal cells. Animal cells don't have vacuoles. And compared with vesicles, vacuoles are a lot larger and contain enzymes that can break down unnecessary macromolecules. Okay, so this is the vacuole right there in a plant cell. Okay, you see how big it is and how much space it takes up inside the plant cell. Okay, it has also other functions. Okay, they also have the capacity to regulate the plant cell's water concentration. Okay, water concentration in any changing environmental conditions. For example, when there are less water around, the vacuole actually shrinks in size, which then will cause the plant to wilt. Or when the plant starts looking droopy because they don't have enough water. Whereas if the plant has enough water, vacuole will be able to enlarge that and looks just like this, okay, and causes cells to expand, which then allows the plant to stand up tall, okay, straight and tall instead of wilty looking, okay. So vesicles, on the other hand, are smaller little pockets or pouches, okay, like this, within the cell that enables the delivery of materials from one part of the cell to another part, or even all the way to be excreted outside of the cell through the plasma membrane. This was the plasma membrane right here. The vesicle can actually fuse together. Well, first of all, it will travel towards the plasma membrane, fuse with the plasma membrane, and dump whatever content it is outside the cell. And this will be a process called exocytosis that we'll discuss in chapter 5. Okay. All right. So vesicles are for transportation of materials such as proteins. Okay, so centrosomes are microtubule organizing center that are found close to the nuclei of animal cells. They're not usually in plant cells, and they contain a pair of centrioles within the centrosomes. Okay, but I won't ask you to distinguish between centriole and centrosome. Okay, usually we kind of refer to them as the same thing. So at least know them as centrioles or centrosomes. So if you go back to the animal cell slide here, animal cell picture. Let me see if I can see it here. I can't see it there, so I'll draw it for you. So if nucleus, the centrioles were present, okay, oh, there it is, okay, right here. Okay, that's the centriole. Models and picture, they tend to be drawn close to the nucleus because they are important for cell division, okay? All right, and let me go back to centrioles. Okay, so centrioles are why we call them microtubule organizing centers, because microtubule are a type of cytoskeleton that is important for maneuvering of chromosomes around the cell during cell division. Okay, which is something we'll discuss later on in the semester. But so centrosomes have some role in pulling the duplicated chromosome to opposite ends of a dividing cell. Let's say if this cell is a dividing cell, what we'll talk about in mitosis is that the chromosomes or DNA will line up towards the middle of the dividing cell. The centrosome are going to be on this side and this side of the cell, and they will have little lines or fibers radiating out from it, and they will sort of attach to the chromosome and pull the chromosome either this way or that way, so that becomes until they will eventually split into two different daughter cells, splitting the chromosomes into two. Okay. We'll talk about that when we get to the next, that chapter. Okay, just remember centrosomes are important for the function of pulling apart duplicated chromosomes in a dividing cell. Other than that, they don't really serve other purposes, not that we know of currently anyway. So lysosome is known as the cell's garbage disposal because they are, as it says, okay, they get rid of stuff that is no longer necessary within the cell. Okay, kind of analogous to vacuoles in plant cells. Inside the lysosomes are usually digestive enzymes that can break down macromolecules or even worn out organelles that are no longer necessary. Okay, and because they serve the purpose of digesting materials, the pH within the lysosomes are more acidic. They tend to be more acidic than the cytoplasms. Again, going back to what we talked about last in chapter 6, saying that, different parts of the cell has to be compartmentalized because we don't want things to be happening where they're not supposed to. In this case, we don't want digestive enzymes to be out in the cytoplasm. Then it will start digesting everything we have. It will digest the ribosomes. It will digest all the rough ER, all those stuff that's floating around for important purposes. Okay, so the lysosomes are what contains those digestive enzymes only for things we no longer need, and they will only digest those stuffs within it. Okay, all right. Let's see, not the last structure yet. Okay, we're close to it. So there is another structure that is only present in plant cells, is the cell wall. Okay, now we kind of discussed and introduced this in chapter 3. And remember that it was a polysaccharide that is made up of cellulose. Okay, it is a rigid covering for the plant cell right here. Okay, so this is the cell wall, this green whole part of the cell. And they are for structural support, okay, of the plant cell that enables their shaping how the cell is supposed to look like. Kind of like what I said in chapter 3, plants don't have bones to support their structure. So what supports them to uphold and stand up high and stand up tall is their cell wall that provides for that structural integrity of the whole organism itself. So as we learned before, the major component in plant cell wall is cellulose. Again, don't confuse this with prokaryotic cells. In prokaryotic cell, their cell wall is made up of another substance called peptidoglycan. Okay, the cell wall for plant cells is made up of cellulose. Okay. But yet, the capsule of prokaryotic cell is made up of polysaccharide, okay? What a lot of students end up confusing and doing on the prokaryotic cell is they would say the cell wall in the prokaryotic cell is made up of polysaccharide and the capsule is made of peptidoglycan. So make sure you don't mix those two up, okay? The cell wall of prokaryotic cell contains the peptidoglycan. It is the capsule of prokaryotic cell that contains a polysaccharide. For plant cell, it is a cell wall that contains a polysaccharide that is called the cellulose, okay, made up of cellulose. All right, so the last structure that is only present in plants, as well as only in plants, not in animals, are called chloroplasts, okay? Chloroplasts, which is the main organelle that gives plant cells their ability to make glucose from carbon dioxide and water with the help of sunlight energy. And this is a chemical process called photosynthesis, okay? And we'll talk about the details of this process in chapter 8. And inside the chloroplast, kind of like mitochondria, they also have a double membrane. The inner membrane will contain stacks of these green disc-looking things. Those disc-looking things are called thylakoids, okay? We'll learn about their function in photosynthesis, but they're fluid-filled membrane sacs that are stacked on top of each other to form a granum, okay? So one of these discs is called a thylakoid. A whole stack of them is called the granum, okay? And the critical molecule this organelle contains that allows for usage of sunlight as energy source is the green photosynthetic pigment called chlorophyll. Okay, if we humans have chlorophylls in our skin, then first of all, our skin will be green and we will be able to go out into the sun and not eat and still produce glucose because we are able to turn sunlight into sugar for nutrient and making ATP and stuff. So, but we don't. Okay, so only plant cells have chlorophyll, which was why they're green. And only parts of the plant that is exposed to sunlight is usually green and has chlorophyll in it, which explains why a plant's roots are not green. Okay, we pull up a plant, the roots is usually white, okay, or yellowish in color, and also explain why onion and ginger from underneath the ground is not green like cabbage or lettuce, is because they are underneath the ground. They're not exposed to the sun, so they don't do any photosynthesis. It's not necessary for them to do any photosynthesis because they can't. They're underground. Okay. All right. So the endoplasmic reticulum, known as the ER for short, is really just a series of interconnected membranous sacs or tubules that serve the purpose of protein modification and lipid production. And the phospholipid bilayer is actually continuous with the nuclear envelope. Okay, so we go from the center of the cell where the nucleus was, and radiating out from the nucleus are these sacs. Okay, and the first that comes out of it is the rough ER, followed by the smooth ER. Okay. So the rough ER, okay, is, and its name rough actually comes from the fact that it has ribosomes studded all over it, okay, and it serves of of course, just like what we discussed, ribosomes are important for. So rough ER does a very important purpose of protein synthesis, the making of protein, and hence this is why rough ER is usually known as a protein factory for the cell. It also makes phospholipids for cell membranes as well. And once the finished protein is produced from the rough ER, it will be packaged in a transport vesicle, kind of what we talked about in what a vesicle was. And it will bud off like this in a little bud, okay, and then travel in a little vesicle to other areas of the cell and move on to their next destination, which usually is the Golgi apparatus. So the next section of ER that is continuous with the rough ER is the smooth ER. Is named having the word smooth in it, tells you that it doesn't contain much ribosome, and its main function is for the production of lipids and carbohydrates and steroid hormones. They also play a role in detoxification of medication, poisons, and toxins, okay, which is why liver cells would have a lot of smooth ER. They also serve as a storage of calcium ions as well that are needed for muscle contractions, okay? All right, so that's the rough ER and smooth ER, okay, and how they look like on a cell. The last but not least eukaryotic cell organelle is the Golgi apparatus, okay, which is a series of flattened membranes that looks just like this right here for sorting, packaging, tagging, and distribution of lipids and proteins to their final destination, okay. And just like a tunnel, it has an entrance and an exit side. Okay, remember I talked about how the vesicle buds off from the rough ER right here. It buds off from the rough ER after proteins are produced and they're packaged within the vesicle like this. And then they can move on to the Golgi apparatus. Here's that vesicle with the proteins within it coming off from the rough ER and is now coming towards the Golgi apparatus right here. And hence, the entrance side is where the package transport vesicle will travel to, okay, so that's called the cis phase, okay, cis phase of the Golgi apparatus, where the vesicle will merge with it, sort of fuse together with the cis phase of the Golgi apparatus, dump the protein contents into the Golgi apparatus, and then the proteins will then move throughout the Golgi apparatus and then exit through the trans phase. So this is the trans phase of the Golgi apparatus right here, and they will bud out in new vesicles with the newly packaged protein out there. Okay, so these proteins are usually, like I said, repackaged, modified, and marked for their final destination and shipment, which is why they like to call the Golgi apparatus kind of like the UPS company for the cell, shipping those final protein products to their final destination. Okay, then they will exit through the trans phase like I was talking about and bud off into other area of the cell or towards the plasma membrane to be exit or dumped outside of the cell via the process called exocytosis. Okay, so this animation video right here will give you a glimpse of how this process takes place as vesicle comes out of the rough ER, and then they will go into the cis phase of the Golgi apparatus, go through all the membranous sacs of the Golgi apparatus, then comes right out in new buds again at the trans phase of the Golgi apparatus, and those proteins will be packaged in new vesicles to move on to their next destination. So it's a really quick video it doesn't have sound anyway. So here's those rough ER vesicles, transport vesicles. Proteins go through the entire Golgi apparatus and then exit out of it in new transport vesicle on the trans phase. Enters at the cis phase, exits exits out of the trans phase. If you forget that on the exam, remember it's in alphabetical order. Okay, C comes before T. So the one with the C is where vesicle first is fusing with, and then trans phase is where the vesicles are budding off. Okay. All right, so cells that have a lot of secretory activity will have an abundance of these Golgi apparatus. For example, immune cells that are in charge of secreting antibodies will have a lot of these Golgi apparatus within those particular cells. All right. And now that we are done with all the organelles of the eukaryotic cell, there are two additional main structures that we have to discuss. Okay, the first one has to do with how the cell is able to keep its basic structural integrity, kind of what we had a glimpse of in the cell video earlier, where there's roads building and breaking within the cell all the time. Okay, so because after all, if a cell is just made up of phospholipids and jelly matrix of the cytosol, they can easily be smushed or smashed or lose their basic structure and break apart. So there needs to be something harder that will keep the structure of the cell together. So hence long fibers called cytoskeleton is necessary inside the cell to function as a couple of different things. Okay, other than keeping the integrity of the cell, there's also for the purpose of transportation. So one, it functions to maintain the cell shape. They can secure organelles in specific locations to make sure that all of a sudden the nucleus doesn't start migrating towards the plasma membrane, or the centriole starts going towards away from the nucleus. And it allows cytoplasm and vesicles to move within the cell. It kind of usually serves as little tracks for those vesicles to move, which is what you saw in the video earlier with a little foot moving across with a huge bag and some foot that was moving across a line. So that line right there, or tracks within the cell, is what cytoskeleton is made up of. And inside the vesicles are all these proteins that need to be transported. It also is the basic structure that enables a cell to move or crawl to certain areas. Okay, so there are 3 different types of fibers in the cells that are for different purposes. Okay, that serves these different functions we just talked about. The first of those are called intermediate filaments, okay, which are actually fibrous proteins that wound together for structural function. This is just serves for basic integrity structural function of the cell. Okay, is not for things to move on it or anything, or for the cell to move. It's not for that purpose. It's just for structural integrity. Okay, so their main function is to maintain the cell shape by bearing tension and anchoring the organelles in place. An example of intermediate filaments we have in the hair, nails, or skin cells is keratin. And they, if we magnify these intermediate filaments, they kind of look like this bundles of different keratin fibers that are bundled together to keep this basic structure of the cell together. The second type of cytoskeleton is called microfilaments, which is actually the thinnest type of fiber we have for cellular movement. They're mainly made up of 2 actin protein strands that are intertwined with each other. Okay, this is what this is showing right here. One of these is an actin subunit, and they connect to each other in actin filaments, and 2 actin filaments actually twirled around each other like that. And usually for skeletal muscle cells, for example, the actin filament will serve as a track for movement of myosin protein, which is a type of motor protein that has to be powered by ATP, like I was saying with skeletal muscle movement. Okay, for white blood cells, which a lot of times require them to crawl towards the site of infection to fight a foreign invader like bacteria, for example, that crawling movement or motion is made possible by the disassembly and a reassembly of these microfilaments within the cell. And hence the last type of cytoskeleton, and is actually the thickest type, are called microtubules. Okay, remember filaments sounds a lot smaller. They're filaments. Okay, tubules sounds like a whole tube. Okay, so they're thicker. So that's a way you can easily remember the difference between microfilament and microtubules. Even though microtubules are the thickest, they are actually hollow tubes around the cell. Okay, and they serve the purpose of resisting compression. Okay, provide tracks for vesicles to move within the cell, and they also pull replicated chromosome to opposite ends of the dividing cell, which is what we said what centrioles are for. Okay, so centrioles is what's going to organize these microtubules, radiating out from the centrosomes themselves, and they will then attach to chromosomes in the center of the cell, ready to pull them apart for cell division to occur. Okay, and just like what you saw in the video earlier, they also disassemble and assemble very quickly, like highways or tracks within the cell. Okay, for those huge vesicles to walk on. And just like what you saw in and as we have already talked about in the prokaryotic cells, there are cells with flagella, okay, for locomotion. And for eukaryotic human cells, the only type of cells that has this similar type of flagella for movement are sperm cells. Okay, so other than sperm cells, there's no other cells in our body that have a flagella that allows for its movement. Technically speaking, our cells don't need to move around on their own. Okay, red blood cells are just floating around in the fluid. The only thing that there's a certain destination for them to swim against the current of is the sperm cell swimming against the current of of the fluid moving within the uterus and oviducts. Cilia, okay, is something eukaryotic cells have for a different type of movement. Now, what flagella does is that it propels the cell towards its destination. For cilia, it actually moves the environment around the cell. Okay, so it doesn't move the cell, it moves the environment. Okay, it's not the cell that's moving when the cilia moves, but the fluid or object in the cell surrounding that moves. Okay, so it moves substances along the outer surface. Okay, and what cilia shapes like is they are usually short hair-like structures on top of the cell. So, for example, this is what's in the fallopian tubes of the uterus. And those cells that line the uterine tubes or fallopian tubes will have these little cilia coming out of it like that. And they are for the important function or purpose of moving the egg along in the fallopian tube and sort of sweep it in the direction towards the uterus. Okay, because the egg cell does not have little arms or flagella for it to move. So what moves the egg instead are these cilia within the lining of the fallopian tube. So what happens is for females, there's the ovaries and there's the fallopian tubes, and then you have the uterus. Okay, so an egg is ovulated from the ovaries, and then it travels into the fallopian tubes and have to travel into the uterus and embed into the lining in order for the embryo to grow if the egg was fertilized. So what enables the egg to move towards the uterus is these little hairs sweeping the egg across. Okay, the other example of cilia lining all over the place is within our respiratory airways. Okay, so, for example, in our trachea, This is a SEM image of our respiratory cells with tons of cilia on top of those cell that lines our trachea. And why is it important that our airways have all these little cilia is because, and when we breathe in, lots of stuff can come along with the air that we breathe in, dust particles, pollens, whatever it is that we don't want in our lungs. So what happens is the cilia kind of trap, along with mucus, is what traps these particles within the air. And the mucus can be moved upwards by the sweeping motion of these cilia, and then it will sweep the mucus up away from our airway into our mouth so we can spit out the mucus, or they can also be sweep up into our nasal cavity where we can sort of blow out all those the mucus looking things when we blow our noses. Okay, so that's what cilia is important for, in terms of the human body. And on the exterior side of the cells, okay, it's usually not a smooth, bare surface with nothing on it. Okay, it has what's called the extracellular matrix. So this is the plasma membrane. On top of that, these are the extracellular matrix where protein fibers would extend outside of the plasma membrane to hold the cells together, forming tissues and allow for communication of some sort. There's also what's called protein receptors on the outside of the cell that will allow for a molecule to bind or activate or deactivate cellular processes from outside of the cell. For example, platelets will travel in our bloodstream like this. Of course, they're not attached to a site yet in the beginning. They're kind of floating in the bloodstream. But if they sense a signal that's being released by clotting factors, the platelet would then come down here. Let's say if there's a cut or an opening in your blood vessel, platelet would sense those little clotting factors and knows that in somewhere needs to be clotted. So it will then travel to the site of injury and starts coagulating all around that cut site and close up that wound or opening in the blood vessel to stop the bleeding. So that's an example of how important protein receptors are. They are for communication purposes. If we need to activate or deactivate something, and in this case, we needed to activate a clotting process, which is why that protein receptor is necessary. Or another classic example is insulin receptors. Our pancreas, crank out insulin for us to, sort of ingest or uptake glucose from our bloodstream into the liver or the muscle cells. Liver for storage, muscle cells for usage. And so the ability of those liver cells or muscular cells to take in glucose into their cells is by having these insulin receptors. Okay, when, for example, here's a cell, here's an insulin receptor, okay, for example, and if an insulin happened to be bound to it, then it triggers the cell to take in glucose. Okay, without insulin, okay, the cell doesn't know to take glucose. No glucose will be intake from into the cell, and the glucose will continue floating around in our circulatory system in our blood, which is why people with diabetes ends up not being able to use the sugars they ingest, and they end up with a high blood sugar levels because either their body don't generate insulin or something's going on with their protein receptors for insulin. Specifically, this binding action is kind of like that specificity we talked about in the enzyme substrate relationship, kind of like that. But because something's wrong with that, the cell does not sense insulin, and hence it does not intake glucose. Okay, so that's another classic example of the importance of protein receptors. All right. And the last topic, the very last structural component this chapter covers are the connections between cells. For example, we talked about how our bodies or organisms are made of cells bound together. But what binds the cell together? After all, our human body is made up of many cells, but yet they are individual cells. There's no glue that glues them together. We can't just put Elmer's glue and glue cells together. So how are they connected to each other? Hence, we will discuss 3 types of intracellular junction that not only bind cells together in our bodies, but also allow for communication between the cells that will serve their purpose for that specific organ. And the first type of intracellular junction is called tight junctions, which are watertight seal between 2 adjacent cells. So if you imagine this is a cell next to each other, tight junctions are right here. Okay, if we were to magnify them into this. Okay, so that's tight junctions. They're watertight, meaning even fluids cannot pass through these junctions. Okay, for example, imagine cells lining your stomach. Remember I mentioned that stomach acid has a very low pH of 1, very, very acidic solution. And other places of your body does not have the natural protection against this acidic solution. And hence, if the stomach lining were made up of cells that leaks fluid in between them, then we're in trouble. Okay, all our other organs in the abdominal cavity would be in trouble. They would be burned alive with acid. So in some parts of the body, this type of watertight junctions between cells are absolutely vital for our survival. So such as that I mentioned in the stomach, lining of organs, and skin. And the second type of intracellular junction is called desmosomes junction, which kind of are like spot wheels, or what you would like to call it anchor. Okay, we go back to that cellular structure we talked about earlier. These were the tight junctions, and in this area right here are the desmosomes junctions. If we magnify that, this is how it looks like. And these anchors is what resides between adjacent cells to maintain sheet-like formations in organs and tissues. So these are kind of like little staples between cells that keeps them together to make up a whole sheet of tissue, okay, that will serve as your heart, sort of layer of your heart, layer of your muscles, etc. Okay, but they are not watertight seals like tight junction. Okay, they're just like little staples. Okay, that staples the cells together. Okay, desmosome junction. And the last type of intracellular junction is called gap junctions, and that's what's going to be important for the communication purpose between cells, because they actually contain channels between adjacent cells that allow for transportation of ions, nutrients, and substances through there in terms of for communication. So this was a tight junction, this was a desmosome junction. This right here magnified is the gap junctions, because they contain actual holes that are produced by protein channels, okay, that allows for things to move from one cell to another. So for example, in the case of cardiac muscles, there needs to be a rhythmic wave conduction of electrical impulse for the heart to beat correctly. And those signals and ions will be able to travel from one cell to the next very quickly through these channels within the gap junctions. Okay, so these represents the gap junctions between cardiac muscle cells that allow for those signals to go across for the heart to pump correctly and beat correctly. All right, and we will end this chapter by watching the cell video I have introduced earlier in the beginning of the lecture that shows all that needs to happen in the cell just to send the right signal and tell a white blood cell that an infection has taken place, and the area requires the rescue of a white blood cell to squeeze through the blood vessels into the tissues to fight off the pathogen. So the video kind of begins with the white blood cells and red blood cells zooming around in the blood vessels, and then it shows you all that's happening inside those cells that tells the white blood cell, hey, you got to come in to fight an infection. It's pretty cool. Just quick 3 minutes. Oh, hold on. So that's the connection between the two layers of cells, and this is on top of the plasma membrane where you have all these things, the extracellular matrix we were talking about that lies on top. And here's a view of the cytoskeleton underneath, on the inside of the cell. Okay, all these things moving around, serving as cytoskeletons inside the cell. Now we dive into the cytoskeleton towards the center of the cell to see what's going on. And now you see all these stuff we were talking about. Intermediate filament, microfilaments. So these are the microfilaments being built across the cytoplasm. Here's something that snips it off. Okay, snips off the microfilament as necessary. Here are the microtubule forming, assembling as little tracks. Here's a microtubule disassembling. And here's that transport vesicle we talked about with the motor protein footy guy moving across of it, in this sense moving away from the nucleus. And you can see the mitochondria to the right here, and the nucleus and the centrosomes. Here's those messenger RNA shooting out of the nuclear envelope through the nuclear pores, ribosomes zipping through that messenger RNA, building the proteins. There's that protein spitting out from the ribosomes. And then the proteins then bind with each other in that quaternary structure we were talking about in chapter 3, floating past the mitochondria. Here's another different type of protein that's made directly inside. And here's these budding vesicles off from probably a Golgi or a rough ER. There's a Golgi apparatus, cis phase, okay, cis phase, and then trans phase, and then dumped out of the cell in exocytosis. That little protein being dumped signals for these protein receptors to lift up, make contact with other signaling stuff on top, okay, lifts up, make contact with this signaling protein, which then the white blood cell senses that and crawls in, squeezes through the blood vessels and to crawl into the site of infection to fight a particular bacteria. So look at all that needs to happen inside your cell every day without you really thinking about anything, and yet all that is happening and very busy within the cell. All right, and the last thing, actually I have one more thing I want to go over before I let you off the video, is the whole pathway of protein synthesis traveling throughout the cell. So here's the nucleus with the DNA in it, okay, nucleus, DNA. Okay, so, and then what comes out of the DNA is rough ER, of the nucleus is the rough ER, okay, studded with ribosomes. So those dots are ribosomes. Okay, and then little vesicles, okay, buds off from the rough ER and moves on to the Golgi apparatus. And if this was the border of the plasma membrane, then this vesicle would then travel towards the cis phase of the Golgi apparatus. Okay, the vesicle then fuse with the Golgi. Protein travels through the Golgi apparatus and then bud off in another new vesicle with repackaged modified proteins. And then that little bud of vesicle will then either go to other places in the cell or go to the plasma membrane, fuse with it to dump whatever contents outside of the cell into the blood, for example, or extracellular fluid within the body. So this sort of illustrates how instructions within the genetic material can travel out. Okay, but what happens is the mRNA molecule can travel into one of those ribosomes on the rough ER, or ribosomes that are floating around in the cytoplasm, and then they are packaged into little vesicle that can go through the repackaging process within the Golgi apparatus, and then they can be are shipped outside of the cell via a process called exocytosis, okay, into the extracellular fluid or bloodstream. Okay, so that's all I have for you for chapter 4. Remember to start reviewing early for lecture exam 2, which only covers chapter 6 on metabolism and chapter 4 for the cells. Okay, it's not a lot to study, and a lot of students find that this exam is straightforward. So make sure you do your part in studying. Still requires time for studying. Okay, all right, and I'll see you next time. Okay.